our next speaker is so highly accomplished that everything I say will be an understatement. So just very shortly, Jordan Smoller, Director of the Psychiatric and Neurodevelopmental Genetics Unit and Professor of Psychiatry at Mass General Hospital, also a Professor of Epidemiology at Harvard School of Public Health, and very importantly, co-lead of the cross-disorder working group for several years. And this is what he's going to present to us. Jordan.
Thank you so much. I'm presenting on behalf of the cross-disorder group, which I co-chair with Ken Kendler. And let me just give you a bit of an update about what we've been doing. And, you know, this has been said a number of times, but there's been tremendous progress. And you've heard today how this particular figure is outdated already, with just scores of variants identified for our clinical categories of psychiatric diagnosis. But we know that those categories themselves have been something of a moving target. And so, you know, just across the diagnostic system from DSM-1 to DSM-5, we've seen this expansion of diagnostic labels, which I think tells us that our diagnostic categories are not exactly stable. And the cross-disorder group is really designed to kind of address some of these larger picture genetic relationships and looking at all of the available data for the disorders we have. And this was the first paper from the cross-disorder group now back in 2013, in which at the time we had about 61,000 cases and controls in these 5 disorders, and identified loci that seemed to be associated with not just one disorder, but sometimes 3, 4, 5 disorders. And that was followed by an analysis that looked at the genetic correlations among these disorders, which we've now seen, you know, expanded and replicated. But, you know, some of this was sort of striking at the time, the fact that bipolar disorder and schizophrenia, which are canonically different disorders clinically, were so genetically highly correlated. And we now know from many studies, people have mentioned already the Brainstorm Consortium study that's looked at a very large range of these kinds of genetic relationships. But we also know that pleiotropy, this kind of notion that a variant or gene can have multiple effects or effects on multiple phenotypes, is really ubiquitous in the genetic architecture of complex traits, including psychiatric traits. So a recent analysis from Danielle Postema's group and I guess it was last year, looked at the level of pleiotropy across the genome, and 60% of genes had pleiotropic effects. We now have this toolkit that's rapidly expanding of ways that we can look at this across disorder effects and pleiotropy, both at you know, variant and pathway levels up to the genotype. And we've also seen polygenic risk score cross disorder analyses. So what I want to sort of walk you through is the latest analyses that have now been published a couple months ago from the cross-disorder group, and that really was a massive effort among the entire PGC consortium. These analyses were led by Phil Lee, who just did incredible work over a many-year period pulling these data analyses together. And this involved more than 725,000 genome-wide data sets across 8 psychiatric disorders. And these were the data sets that were included in the analyses. Many of them are ones that you've already heard about today. And the first thing that we did was demonstrate again that there is substantial genetic correlation among pairs of disorders. And this sort of network just shows you the level of correlation with the thickness and darkness of the lines representing the strength of that genetic correlation. And bipolar disorder and schizophrenia are closest there on the line as well. But we were able to then make use of another method, Genomics SEM, developed by Andrew Grotzinger, Michel Navarre, and Elliot Tucker-Drobe, that allows us to model the covariance structures among these disorders and fit models to determine what's the best explanation of the genetic relatedness of these pairs of disorders. And what emerged from this was a 3-factor model. There seemed to be 3 factors of genetic relatedness. This was also confirmed in a hierarchical clustering analysis. And the first, which you see on the left there, are disorders that seem to be characterized by compulsive behaviors as a key component. So that was anorexia nervosa, OCD, and Tourette syndrome. The second were the mood and psychotic disorders, schizophrenia, bipolar, and depression. And the third were more early neurodevelopmental disorders, autism spectrum, ADHD, Tourette, and also MDD. So that was contributing to that factor as well. So suggesting that there is some structure among these correlations. The genome-wide meta-analysis putting all these disorders together identified more than 100 loci that were pleiotropic in the sense that they were affecting more than one disorder, and some of them substantially so. So we'll talk in a moment about these two, DCC and RBFOX1, which sort of topped the list in the range of disorders they were associated with. And what you're looking at there is on the top there, DCC, deleted in colorectal cancer, which is also known as the Netrin-1 receptor. And this is a forest plot of the associations for each of those disorders. And then on the right is a PM plot. This is basically a posterior probability estimate of how likely this variant is to be associated with each of these disorders. And so when that exceeds 0.9, we sort of call it as associated with that particular phenotype. And these are interesting genes because they illustrate a couple of ways in which loci can be potentially pleiotropic. DCC turns out to be very centrally involved in axonal migration and essentially the establishment of white matter connections early in neurodevelopment, but also later mediating connections between medial prefrontal cortical dopamine circuitry. And so one could imagine the way in which a variant or a gene like that could be involved in a broad spectrum of earlier and somewhat later onset disorders. RBFOX1 is importantly related to cell type specific alternative splicing kinds of processes that are involved in a whole range, again, of important neurodevelopmental kinds of effects. And so we can imagine how pleiotropy might emerge in those kinds of situations. It turned out that there were 23 loci that were associated with at least 4 different disorders. And this is a table from the paper in Cell, which shows you for those variants, the genes that we were able to map them to based on a convergence of evidence and the number of disorders on the right-hand side that they were associated with. So pointing to some interesting biology, I think, in a number of directions, which we won't have time to go into, obviously. But another feature that emerged was that some of these loci had opposite directional effects on different disorders. So not simply conferring risk to multiple disorders, but in some cases conferring risk or having a protective effect. And sometimes that was in the case of loci affecting disorders that at a genomic level we know are highly correlated. So here's just a couple of examples. A variant that's in the middle there. Actually, that's take the one on the right. That's a locus that's associated with depression and bipolar disorder, but in opposite directions, even though those two disorders are highly correlated. We also, through functional genomic and gene set and pathway analyses, found that not only were many of these top loci involved in neurodevelopmental processes, but at an aggregate level in pathways that were also enriched for neurodevelopmental processes. And also 2 specific neurotransmitter pathways, so glutamatergic and calcium channel signaling genes seem to be enriched in these more broad spectrum phenotypes. And then using gene expression data from postmortem brain, it appeared that the loci that were most pleiotropic showed heightened expression beginning in the 2nd trimester of prenatal life, and then constitutively seemed to be more highly expressed. So these are the lines with the sort of darker blue. Those are the ones associated with more than 2 disorders, and the medium blue there is 2 disorders. There was marked enrichment of these kind of pleiotropic genes in frontal cortex, and particularly in cortical glutamatergic neurons in layers 2 through 6. So we're able to get a little bit of purchase on cell types for which these pleiotropic loci may be most relevant. So to conclude, we see genetic correlation among these 3 groups, among these 8 disorders rather, into structure that gives us a sense of at least 3 groups of highly genetically related disorders. One thing that was notable there, I think, is that there wasn't overwhelming evidence for a kind of overarching genetic single factor liability. But those studies are now expanding in scope already, and we should have new results soon. There were 109 of these loci, some of them highly pleiotropic, and those pleiotropic loci seem to be particularly important, again, in neurodevelopment. One implication of these kinds of loci might be that, for example, in therapeutic development, targeting specific genes or pathways that have particularly pleiotropic effects, on the one hand, they may have broadly therapeutic effects if they're, potentially modulating pathways that are relevant to a number of disorders. But also, as we noted, there are some opposite directional effects that could at least conceivably point us toward anticipating off-target effects or adverse drug effects. We sort of come to the notion that maybe there are at least 2 classes of variants or genes that contribute to psychiatric disorders. So this class of sort of broad liability genes that might be involved, particularly in neurodevelopment and how the brain is essentially wired. And then this much larger, perhaps, set of genes and environmental influences that sort of canalize expression of this vulnerability into some of the discrete syndromes that we recognize. And so the cross-disorder group is continuing to work and include an expanding range of disorders. We did not include all of the disorders that are available. And also to begin a set of analyses, which we think will be very interesting, that will try to dissect the disorder-specific and cross-disorder effects that underlie the genetic architecture of these broad range of disorders. And so that's going to involve the sort of combined application of a number of different methods, which again, no time to go into that at this point, but this is an active area of development for the group. And as has been mentioned previously, it's going to be absolutely crucial to expand this kind of work into more diverse ancestries. So I want to just end there by highlighting a small number of the people who have contributed to the work of the CDG. Phil, I mentioned, and many others who are on this list. The cross-disorder group is listed there, as well as the full Psychiatric Genomics Consortium, iPsych, the UK Biobank, and so many more. And I think I will stop there. I have no idea if we went over or under, but I'm happy to take questions.
Thank you, Jordan. Really exciting work. There's one question that has come in that is related to how does PGC address sample overlap and cross-disorder approaches? Yeah,
that's a great question, and this was the source of a lot of work and angst in the analysis, because one of the things that we have typically done is try to the extent possible, deduplicate and remove at the individual locus level overlapping samples. And we did that here. There's a number of ways that we can do that using checksums and things like that that allow us to get to as little overlap as we can. And many of the methods that are used can sort of account for, particularly some of the genetic correlation methods like LD score regression and so on, can account for this or not substantially biased. The issue of comorbidity is a hugely important and interesting one on its own, because to some extent, you might imagine if you had a lot of comorbidity, you could falsely appear to have genetic relationships between two disorders because your cases are comorbid for those disorders. And that's an interesting conceptual question, because on the one hand, you wouldn't want to exclude comorbid cases. If there is, in fact, genetic relationship, you expect some degree of comorbidity. But on the other hand, we were limited in any case in this setting because only a subset of cases really had comorbidity. We did a number of analyses to try to address the robustness of that, but it's an interesting potential challenge for the field, I think. Yeah,
I think so too. I have another question regarding the genomic SEM, and that is, I was surprised to see that MDD is clustering together with ADHD and ASD and Tourette, I think.
Yeah.
How do you think that comes along?
Well, what we speculate, although without any real data behind it exactly, is that MDD itself is likely one of the more heterogeneous disorders. And we know, for example, that there are folks who have early onset MDD, which typically has a more persistent course and a more severe course. Sometimes is more related to bipolar disorder, as a matter of fact. And so we may be seeing that kind of stratification within the phenotype in which some of the loading ends up on the neurodevelopmental side. There have been some further genomic SEM analyses that are now ongoing with the addition of additional disorders, which suggests that we may see a little bit of reshuffling of MDD, but that's the best we can come up with at this point.
Okay. We're getting a lot of messages here that there is a talk on Friday on genomic SEM that will go more into that. Okay. And that after this session, we will also have the possibility to go to different rooms where you can ask more questions to the speakers.